Today I'll present our work using focused ultrasound disruption of the blood-brain barrier to deliver molecular therapies to the brain. And this project is supported by the Focused Ultrasound Surgery Foundation and is being done in collaboration with Moderna. So in our wider project, we're looking at a class of diseases known as lysosomal storage disorders, where typically a single gene mutation leads to a loss of function that affects lysosomal function. And this is then characterized by an accumulation of toxic metabolites in cells, such as cholesterol, lipids. And while individually rare, there are over 70 different lysosomal storage disorders that collectively affect a large number of patients. And importantly for our work, most of these have some component of neurodegeneration involved in the disease. And within this particular project, we're focusing on the lysosomal storage disorder known as NeimanPick C, which is a loss of function due to a mutation in the NPC1 gene that affects the lysosomal membrane and leads to an accumulation of cholesterol in cells. And the disease is known to have a number of different neurological manifestations, such as ultimately cognitive and psychiatric impairments. And one particular promising approach is to use a messenger RNA therapy to exogenously introduce the NPC1 protein and restore the loss function of the lysosome. So our goals are to first show that we can use this therapy, which has been provided by Moderna, to translate into the NPC1 protein and therefore reduce cholesterol accumulation in cells. And crucially for us, we have to be able to deliver it to the cerebellum where the disease is most manifest, and then show that that will restore cell health. So our first step was to validate the therapy first in cell lines, and our collaborators at Boston Children's Hospital looked at 3 different formulations, each encapsulated in a lipid nanoparticle, and 2 no therapy control groups. And you can see on the plots measuring the level of cholesterol storage in these cells. Baseline in normal cells was not affected over the 5 different formulations tested. When they looked at NPC1 cells that have the mutation, the 2 control groups on the ends there show significantly raised levels of cholesterol storage. However, each of the therapies was able to bring that level back down to baseline. So in these cell lines, they were able to show that, in fact, this therapy is translated into the NPC1 protein, and it does reduce cholesterol accumulation in those cells. So the next step was to try to validate the therapy in vivo, again using a lipid nanoparticle formulation and injected IV, first in control mice and then an NPC mouse model that's homozygous for the mutated protein. And also looking at those 3 formulations and the 1 control that's just GFP. So what you can see on these Western blots taken from liver, spleen, and cerebellum, in the wild type mice, as we would expect, they have a large level of the protein present because they don't have the mutation in all 3 organs looked at. When just giving the sort of no therapy control, the eGFP, that does not lead to an accumulation of the NPC1 protein in any of the NPC mice model because the mutation means that it's totally absent. However, when we give the therapy that has the mRNA for producing the NPC1 protein, we see in liver and spleen a big uptick in the amount of protein present. However, when we look in the cerebellum, we don't see any protein, indicating that the therapy is reaching peripheral organs but not penetrating into the brain. And this confirms some of these findings looking at liver sections under an H&E stain and imaging, where the top is the sort of what we would expect from the wild type mice, a healthy control. And then the second panel is showing an NPC mouse model where these enlarged cells are due to additional cholesterol accumulation. And then when we treat with the therapy, a lot of that is reduced. So now in vivo, we've been able to validate that we get both translation of the protein and enough protein delivered so that we reduce cholesterol accumulation. So now we wanted to go on and validate delivery to the brain. So our first step was to do that in a wild type mice, and we're targeting focused ultrasound to the cerebellum for BBB disruption. And again, giving the lipid nanoparticle, in this case, just with GFP by tail vein injection. And you can see over these 5 mice that we get reasonable BBB disruption in the cerebellum, as indicated by contrast MRI. And then when we did Western blot on the cerebellum controlling or looking at the focused ultrasound groups versus the no focused ultrasound groups, there's a significant increase in the GFP level in the FUS treated groups. So now we know that we can deliver this lipid nanoparticle with the mRNA formulation to the cerebellum. And so the last phase of the study is to then look at actually delivering the mRNA therapy for a therapeutic efficacy study. And for this, we're going to use NPC model mice, still target focused ultrasound to the cerebellum. But then I think interesting to use 2 control groups. One is the standard no FUS control group, but then we're also going to add a positive control group where we're delivering the therapy by cisterna magna injection, which would be kind of equivalent to a intrathecal injection in the human and getting it into the CSF space. And we're going to look at outcome measures for both the distribution of the therapy within the CNS and also some markers of therapeutic efficacy, such as the protein translation and the level of cholesterol, and then some more markers of kind of cell health around inflammation and cell morphology. So we've done the 2 no FUS groups where we've given the therapy by retroorbital injection, and we're about to start the FUS therapy groups, and we hope to have those results soon. So thank you everyone for listening, and thanks to all our great collaborators, in particular Moderna and the Focused Ultrasound Surgery Foundation. Thank you.